Can you really grow a brain in a Petri dish? It sounds like something straight out of a sci-fi movie, but what if I told you it's happening right now in labs all over the world? This isn't science fiction, it's the cutting edge of neuroscience, and it could change everything we know about the human mind. So how is this possible, and what does it mean for the future of humanity? Stick around because we're diving deep into the incredible and slightly unsettling world of brain organoids. Let's start at the beginning. How do you even begin to grow a brain? It all starts with something called pluripotent stem cells. These are amazing cells that have the potential to become any type of cell in the body. Scientists take these stem cells, often from a simple skin or blood sample, and place them in a special nutrient-rich broth inside a Petri dish. It's like a five-star hotel for cells. They provide the perfect conditions, the right temperature, the right food, and the right chemical signals to gently persuade these blank slate cells to start a new career path, becoming brain cells. Think of it like giving a block of clay to a sculptor. The potential is there, but you need to guide it. Scientists use a specific cocktail of proteins and growth factors, essentially whispering instructions to the cells. They'll say, okay, we need you to become a neuron, or you're going to be a glial cell, the support system of the brain. It's a delicate dance of biochemistry, a recipe refined through years of trial and error. The dish is kept in a bioreactor, which gently spins to ensure all the cells get equal access to nutrients and oxygen, mimicking the environment of a developing embryo. And then, the magic happens. Under the microscope, you can see these cells begin to divide and multiply. What starts as a flat layer of individual cells begins to organize itself. It's almost like they have an innate blueprint they're following. They start to clump together, forming tiny, spherical structures. These are the first seeds of a brain organoid. It's a slow, meticulous process, but within just a few weeks, these tiny clusters, no bigger than a pinhead, start to show remarkable complexity. As these organoids grow, they don't just become a random blob of brain cells, they self-organize. You can actually see distinct regions forming, mimicking the early stages of a human brain's development. Layers form that resemble the cerebral cortex, the part of our brain responsible for thought, language, and memory. Under powerful microscopes, you can witness neurons sending out long, spindly arms called axons, reaching out to connect with other neurons. They form intricate networks, creating synapses, the very connections that allow our own brains to process information. These little organoids are building their own neural circuits, right there in the dish. It's not a full-size brain, of course, but a miniature, simplified model that holds immense power. So why go to all this trouble? Why grow mini brains in a lab? The impact on medicine is staggering. For centuries, the human brain has been a black box. We can't just open up a living person's skull to study diseases like Alzheimer's, Parkinson's, or schizophrenia in real time. Animal models, like mice, are helpful, but a mouse brain is fundamentally different from a human brain. Brain organoids change the game completely. Imagine you could take skin cells from a patient with a specific genetic neurological disorder, turn them into a brain organoid, and then watch that disease develop in a dish. That's exactly what scientists are doing. They can test thousands of potential drugs directly on these human-like brain tissues to see which ones work, which ones don't, and which ones have harmful side effects, all without ever putting a patient at risk. This allows for personalized medicine on an unprecedented level. We can create a disease in a dish model for an individual and find the perfect treatment for them. It's also revolutionizing our understanding of brain development itself, helping us figure out what goes wrong in conditions like autism or Zika virus infection. But as with any powerful technology, this raises some profound ethical questions. The headlines are already buzzing with debate. We're creating structures that are biologically human, that have neural activity, and that are patterned like our own brains. Where do we draw the line? People are asking, is this playing God? And should this even be allowed? The core of the debate centers on one terrifying, fascinating question. Could these organoids ever become conscious? Let's be clear, right now, these organoids are not conscious. They lack the size, the structure, the sensory input, and the connection to a body that are all essential for the kind of consciousness we experience. They are tiny, isolated clusters of cells, but the technology is advancing at a breathtaking pace. Scientists have already detected coordinated waves of electrical activity in more complex organoids, patterns that look eerily similar to the brain waves of premature babies. This has sent a shockwave through the scientific and ethical communities. Simulations of this brain activity try to model what's happening inside. Are these just random electrical crackles, or is it the beginning of organized thought? Well, the consensus is that it's not thinking in any meaningful way. It forces us to confront a future where it might. Ethicists are now working alongside scientists to establish clear guidelines. How complex can we allow these organoids to become? At what point would we need to grant them some form of moral status? These aren't easy questions, and they challenge our very definition of what it means to be alive and to be human. So, what's next on the horizon? The future of organoid technology is both exciting and a little bit mind-bending. Researchers are working on making them even more complex. They're developing ways to add blood vessels to the organoids, allowing them to grow larger and live longer. They're experimenting with connecting different types of organoids together. For example, linking a brain organoid to a retinal organoid that can detect light to see how they communicate. Some labs are even exploring the idea of connecting these organoids to robotic bodies or computer interfaces. It has a field moving so fast that what seems like fantasy today could be a laboratory reality tomorrow. The goal is to create ever more sophisticated models to unlock the deepest secrets of the human brain, from devastating diseases to the very nature of our thoughts and emotions. We stand at a unique moment in history, peering into a glowing dish that holds a miniature model of our own mind. The journey of the brain organoid is just beginning, and it promises to take us to places we've only ever dreamed of. It forces us to ask big questions not just about science, but about ourselves. What we learn from these tiny brains could one day save millions of lives and redefine our understanding of existence itself. Thank you so much for joining me on this incredible scientific adventure. If you're as fascinated by these groundbreaking experiments as I am, make sure you subscribe and hit that notification bell. We have so much more to explore together. See you in the next one.